Good morning. I am a cornea surgeon, and I spend a lot of time in my career to do all forms of cornea transplants, DMAC, DSEC, DEL, PKP, and corneal surface reconstructions. Although it's very gratifying to see these patients regain vision after a successful surgery, blinding complications can occur, and cornea transplant does not last forever. Therefore, I devote a significant amount of my time with my team to develop new therapy or better therapy for corneal blindness. Corneal blindness is the fourth leading cause of blindness globally. Every year, there are 1.2 million new cases, in addition to 12 million cases who are already blind. Corneal stromal scar is the leading cause of corneal blindness. In the world, because of a shortage of corneal tissue, only 1 in 70 patients will receive a cornea transplant in their lifetime. The maintenance of the cornea transparency is by 3 layers: epithelium, stroma, and endothelium plus the descemes membrane. Each structure provides a certain function to maintain the clarity of the cornea. So in my laboratory, we first embarked to develop a autologous Limbo stem cell therapy to treat Limbo stem cell deficiency, as seen on the left side. And this is a blinding disease and will also cause significant pain and photophobia in the patients. Cornea transplant does not treat stem cell deficiency. We developed a xeno-free and feeder-free system to manufacture these stem cells from a biopsy, and this is the end product you can see. So we just cleared by the FDA to initiate a phase 1 clinical trial to treat this orphan disease. And a biopsy can be obtained from the donor eye, culture them in the dish, and then transplanted to the diseased eye. If the eye needs a cornea transplant, you see the last panel, that can be performed and can achieve much higher success. So we also develop a new imaging system to visualize these stem cells in live patients. So we are going to validate this new method to look into the outcome of the clinical trial and also to detect any residual stem cells in eyes with partial Limbo stem cell deficiency, and hopefully we can manufacture them and treat patients with bilateral disease as well. The second project we have been working on is a cell-free treatment for stromal scars that is carried out in collaborations with Dr. James Vandenberg at Pittsburgh, who discovered a group of stromal stem cells in the Limbo region. And these stem cells can reduce scar formation in humans. As you see in the last panel, this patient, before the treatment, is significant scarring, and after treatment, and a corneal transplant is not necessary. More excitingly, we found that extracellular vesicles from these stem cells can reduce scar formation as well. So this is the in mouse model, and the result is very striking. We're able to generate immortalized line, and more excitingly, we found that the VVs from these immortalized cells can reduce corneal scar as well, as good as the parental lines, the cells. So that's what we want to develop for the future for corneal stromal scars. We isolated EVs from these immortalized cells, package them as a off-the-shelf product, and apply them topically on the cornea and to reduce scar formation instead of a corneal transplant. We have 2 posters today. One is on the Limbo stem cell therapy, which will be presented by Sheila Gonzalez. The second poster will be presented by Dr. Dos Santos on the cell-free therapy for stromal scars. And I welcome partnerships from industry to move these 2 therapies to the clinic. Thank you.